Hello everyone, I'm Duygu and welcome to the Nano Open webinar organized by Sabanc University Nanotechnology Research and Application Center, which is called Sunum. Today, we'll talk on how nanotechnology is transforming medicine and the future of biology with Professor Sonia Contera. Before getting started to the session, I have some reminders for you. Firstly, all attendees will be in listen-only mode. Secondly, the session is being recorded. And finally, if you have any questions during the webinar, please type them into the chat box. We'll also have time for questions at the end. Well, here at Sunum, we are working on tiny issues in terms of size, but mighty. Before diving into today's topic session, I'll pass over to my colleague, Professor Dr. Fazilet Vardar Sukan, who is the director of Sunum. Professor Vardar, could you please give a brief introduction about Sabanc University Nanotechnology Research and Application Center?
Well, first of all, I'd like to thank Dr. Contera for accepting this. It's a very nice opportunity for everyone, and we're all very excited to be hosting her. I don't want to steal the audience, so this will be really very brief. Sabanc University Nanotechnology Center was first established in the year 2010 with investment from Sabancı Foundation and Turkish State Planning Institute. With investment of $35 million. And in the year 2017, it was chosen by the Ministry of Development as one of the four national infrastructures modeled after the Fraunhofer Institutes. So we are basically aiming to be a bridge between basic research and industry and trying to create socio-economic value throughout research results. The vision of the center is a Turkey that generates knowledge and added value in nanotechnology. So that is our main emphasis. We have, the building is very unique. It is modeled after a biological cell with a special architecture. And it is a two-story building with approximately 8,000 square meters. We have a clean room, which is probably one of the best in the country. Currently, there are 120 people working under this roof. 100 of them are researchers, postgraduates, postdoctorals, and so on. And we concentrate our research topics depending on our institutional strengths, national priorities, industrial needs, and international trends, because it's very important for us to be also in the cross-cutting level in the global arena. And the topics that we concentrate are nanomaterials in the horizontal, micro nanosystems design and integration, and nanomaterials production and characterization. And we have four vertical pillars: life sciences, food, agriculture, water and environment, energy applications, and defense, aviation, and aerospace. So these are the basic areas. We have a number of strategic partners from industry, from academia, over 200 strategic partners currently we are working with. And as a last word, I'd like to emphasize that Sunum follows the unique example of Sabancı University for multidisciplinary application-oriented research with its creating and developing together strategy and no departments, no walls approach, complementing the research expertise of different internal units and external stakeholders. And we are open to collaborations globally with academic and industrial partners. We are now very happy to be hosting Dr. Contera and excited to be hearing about her presentation. Thank you.
Well, thank you, Professor Vardar, for this great brief introduction. Now, I'm absolutely delighted to welcome here one of the world's leading nanotech pioneers, Professor Dr. Sonia Contera. Sonia Contera is Associate Professor of Biological Physics in the Department of Physics at the University of Oxford and author of the book called Nano Comes to Life: How Nanotechnology is Transforming Medicine and the Future of Biology. Let's see what's happening in this field of nanotechnology and how they are coming to our life. Professor Contera, first of all, I'd like to ask you, what are nanometer and nanotechnology? Could you please explain them with some examples briefly?
Yes, a nanometer is important because it's the scale of our proteins, of our molecules. It's the scale that physics, the universe, chose, for reasons I will discuss a little bit, to create life on Earth. It's a very special scale. It's 1,000 times smaller than a micron, so it's very, very small. But is this the importance of this scale is that you can couple mechanics, chemistry, electricity, all in this size. That's why life emerged, and that's why it's important to understand biology, to create future technologies that are inspired by the power of biology, and also to improve medical treatments and indeed detecting things like pathogens and viruses.
Yeah, well, thank you. I know that you were actually a physicist, and then you focused on nanotechnology and then biology in different countries like Spain, China, Denmark, and finally you are located in the UK. It really seems inspiring journey. Could you please explain how does physics change the study of biology at the nanoscale? And also, I wonder the relationship between biology and nanotechnology.
Yes, as I said at the beginning, the reason why mainly nanotechnology is important is because the size of our molecules is a nanoscale. And that brings a lot of fascinating physics and a lot of applications in medicine and a lot of questions about ourselves. So I'm a physicist, and physicists were the first ones that produced the tools to study matter at the nanometer scale. As I will review in my talk, I do a little bit of historical introduction. And it was the interest in matter at the nanometer scale that brought us to biology. And that is one of the arguments of my book, and I will discuss today, and why nanotechnology is important. It breaks barriers between disciplines. It brings physicists, material scientists, computer scientists, biologists, medics together. And as we are seeing now, and as we know when we talk to doctors, in the main medical problems we have in our world, let's say cancer or detecting pathogens or curing diseases, we need a multidisciplinary approach. Simple reductionist approaches based just on molecules have not solved the problem of cancer, and we need a more integrated approach. And this is why I think nanotechnology is important, because historically it's been a main contributor to breaking barriers between disciplines.
Mm-hmm. Well, at this point, I'd like to deep dive into your words. I mean, how can nanotechnology be used to address the medical problems? I mean, for example, as you said, is it possible to improve the efficiency of current cancer chemotherapies in nanotechnology? How we can do that?
Well, yes, but so far nanotechnology has failed. And this is what I'm discussing in my talk, why it has failed. But I think we have hopes of having learned the lessons of pharmacology and nanomedicine failing, and create new strategies that are more integrative of biology as a whole, that just don't look at molecules, but they look at the whole system's biology to improve treatment. So in that sense, nanotechnology is important because it brings tools to understand and tools to deliver drugs. Yeah.
Well, now maybe let's look at the future and focus on transformation on biology and medicine that you mentioned in your book as well. How medicine and health expect to benefit from nanotechnology, and what will be the most stunning contributions of nanotechnology to these fields?
I will review that in my book. I think there are concrete areas such as biosensing, tissue engineering, drug delivery that will benefit directly from nanomedicine. But I think, as I keep telling, the most important part is that it brings other people to look at biology, and we can create new interfaces with other fields, such as, for example, information processing, machine learning. We are in a moment of history where we are starting to understand how we think, why biological matter is structured from the nanometer scale up can understand the world around us. That will bring a revolution in materials. We will start to understand how to create materials that are smarter. They will have applications in medicine, and it will also bring a revolution in the way we understand ourselves. So I think we're in a moment of change in which the nanometer scale and nanotechnology is central, because it's the scale from which life is made. There are very profound physical, why the universe created life at that scale, and also philosophical questions. And I think it's important to discuss the history, and in even the philosophical context of the things we do, because this will allow us to produce better technologies, not because they are effective, but they're more useful for our societies. And in that sense, I think nanotechnology is an important example for its development, is history and the reception it has had in the sciences and in the public.
Yeah, well, I know that your presentation, maybe you can, we can see your presentation, and I would like to ask that point, what was your main motivation or purpose to write the book called Nano Comes to Life?
Because there's no, since Eric Dressler worked his first book on nanotechnology in the 1980s, there's been no real intellectual reflection of the development of nanotechnology and where it's leading us. As physicists, our scientists, our medics, we're too busy doing our jobs trying to survive, and we don't reflect on the consequences and the breaches that our technologies and our knowledge is making. We leave this job to philosophers, to historians. The problem I have found in the books that have become international bestsellers and talking about technology, they don't really know the technology. They don't know the cutting edge. They don't know how scientists do technology. They assume what are our motivations. I wanted to bring the point of view of a multidisciplinary scientist, a woman from Spain that has traveled all over the world and has seen a lot of things. And I wanted to reflect on the future of technology from a scientist point of view. I think we don't do enough reflection as scientists. We don't engage enough with the public conversation about how we apply science. We're too busy fighting the politics of our department just to get grants and to keep going and trying to convince governments to give us money. But I think the world is facing much more complicated problems, and we have to become more public scientists. We need to work for our societies, and we need to work together with scientists of other countries. That's why I wrote the books, to initiate conversations.
You're totally right. Well, maybe we can move further with your presentation, if it's okay for you, Professor Contera.
Of course. So I'm going to share my screen. I hope you can see it. Bear with me. Oh my God, it's not sharing. Is it sharing now? Yes.
Yeah, we can see.
Yeah, we are. So as your questions have given very much prelude to my talk, I'm going to cover some of the things you've said before. I'll try to keep it to my 45 minutes. As you say, I like talking, and I think a lot. I've been thinking a lot because I've been in lockdown, so I'm even worse than used to. I'll take a little bit an overview of nanotech, a little bit historical, a little bit the areas of medicine that I think it will be more important to continue doing science, and I do a little bit about my research in the end, if I may. Okay, so let's go a little bit historically. In the 1980s is when nanotech started, and we started to see, so before that, in the 1950s, Richard Feynman famously said, if we manage to reach the nanoscale, we will unlock a whole world of manufacturing and applications that we never had before. I don't think he realized how important it was, the nanotech, but in the 1980s, scientists were starting to actually reach, with experiments, not just with theories, the nanoscale. So the first scientists that started to publish in the area are Harry Kroto, and Smiley, that published the papers about the discovery of the fullerene, which is still around and is still something we probably will be using in nanomedicine at some point. It's a little buckyball made of carbon atoms 1 nanometer across. Also in the 1980s, something interesting happened, and because I talk a lot about the interface of nano with biology, let's sit a little bit here. So Nagant had spent most of his before he was a professor in New York, he was working for a long time trying to understand the structures of DNA from crystal structures. He was actually working for many years trying to understand DNA from Xray diffraction experiments. When he got his position in New York, he didn't get grants, he was doing very badly, and he started to think about if it was possible to use DNA as a building block. He's the first person who thought about it. And I think the reason why he thought about it is because he was used to see DNA as a building block of crystals, not as a part of the cell, but as something he had to use as a structural material. And he was the first one to be able to construct, to design in a computer, a little bit primitively at the beginning, DNA strands that will able to stick to each other and form shapes. This field has now evolved into an international field. It still has not delivered much applications. As I tell later, we're starting to see the first applications of DNA nanotechnology later. So this also happened in the 1980s. In the 1980s, the famous book, Edith Dresler, Engines of Creation, he was thinking, he was a scientist thinking, and he wrote a book. Imagining how the future could be. I think, and I will tell later, he made a few mistakes with the nanorobots, but it was great that he imagined them. We have nanorobots now. They don't look like any Dresler nanorobots, and we have created it in very different ways, and this is something I will discuss later. And for me, the key was this, the discovery of the scanning probe microscopy, an area where Turkey is very strong. You have scientists abroad and in Turkey, which are leaders in this field. So I'm very happy to bring them here because they made massive contributions to the development of our area. So in 1980s, these guys in Zurich created the scanning tunnel microscope, and it was before them you could not see atoms. You can see matter below hundreds of nanometers. You could just get X-ray diffraction images, which was like difficult, a lot of analysis in form. So the scanning probe microscope does not use light, does not use electrons, or it does not use a wave to see the sample, but it uses a nanotech concept. It uses a very sharp tip, which is an atom thin, to scan it over the surface. And by the interaction of the tip and the sample and a computer program and physics, you can produce images of surfaces like this one. So what you see in this picture is a background of copper atoms. And what you see here at the top are iron atoms that have been deposited on the sample. But because it's not wave, but you have a little finger there touching on the surface, the scanning probe microscopes allow you to interact with matter at the atomic and the nanometer scale. So what this guy did, this is Don Eigler in IBM. They constructed an STM in the 1980s. And in the 1990s, he showed this experiment, which I always like showing because it's the first, I think, that shows the power of nanotech. So what he did was to pick up the atoms of iron. These are no pictures, this is microscopy, this is real, and arrange them in a circle. This is the first. It's the first time humans construct anything, atoms one by one. This is not a very practical way of constructing things, but it shows the proof of principle. Humans can construct with atoms. And it also shows you the mindset of science. We are very reductionist. We like looking into the building blocks of things and then arrange them together intellectually with our theories or physically like he did here. But it also shows you the response of nature. Okay, you put your atoms in a circle, but look what happens here is a ripple coming up from the surface. Where does it come from? These are electrons. The electrons of the copper are confined by the atom of iron, and they can leave, and they behave like a fluid. You can see the wave there. It's a beautiful visualization of quantum mechanics. But it also tells you something very important that I will tell in my talk today. As humans, we are used to develop technology and to think about nature, also in medicine, in a very reductionist way. We think about building blocks, atoms, molecules, DNA, and we want to fix them, arrange them to create things. But nature doesn't work like that. What we have learned in the 21st century is to learn about nature's complexity. Even in this very simple system, what we find is that the strong interaction between electrons and atoms in a crystal creates phenomena that cannot be easily explained by the interaction of the parts alone. In nature, more is different. There are things happen with things interact with each other complexly with feedback loops that we have in biology that gives us behavior that we cannot understand just by knowing the building blocks. This is the main fundamental intellectual idea I want to pass in this talk. So here's the atomic force microscopes. And what people like me, I was doing my PhD in the 1990s in Japan. I was interested in nanotech. I was interested in these microscopes, but I started to get very interested in medicine, in biology. Why? Because as I said at the beginning of your questions, biology is made of proteins and DNA and biomolecules, and they all have nanometer scale. And what we were started to learn in the 1990s is that these proteins were not just boring little blocks that didn't move because they're nano and they're in hot water. What biology had managed to produce and the way life emerged on Earth is by creating amazing nanomachines. So in the 1990s, we were starting to have a glimpse of these nanomachines using, for example, Xray diffraction or other techniques, but we didn't know how they work. We didn't know the physics of how they move, how they use temperature. And our techniques, and this is where I became interested in nanotech, would allow us for the first time to understand the physics, how do they work, the reverse engineering of proteins. So we started to move from cartoons like this ones to actual movies, microscopy of what was going on. And we have our new microscopes that they could poke and pull at the nanometer scale. So I was inspired by the work of Hiroyuki Noji and others in Japan that were started to understand, for example, these proteins like this one. This is the ATP synthase that gave a Nobel Prize in chemistry in 1997 to Paul Boyer, John Walker, and Jens Skov Pedersen in Denmark. These guys were in England. What this thing does, you have kilograms of this in your body, and this is the example of nanomachine I'm going to tell you, biology, is sitting in your body all over producing ATP. So the molecules in our body are not produced like in a test tube. That will not be efficient. It's not like our chemistry. They have amazing catalytic systems to produce really fast chemistry when we need it. So what do they do? Look at this one. This one is sitting in the surface of our membranes. Our cells are covered like a bag, and this bag is called a membrane, and it has lots of nanomachines sitting on it, lots of membrane proteins. So this one collects protons from one side, and by a combination of mechanics and electrostatics is able to create a rotation movement like a motor at the bottom, transmit the rotation through the shaft, and create a movement at the top. So it's like a two couple motors moving at the same time, just using mechanics and electrostatics. And the interesting thing is that this top part is able to buy ADP, binds it, twists it, spits it. And doing that is very efficient at creating a chemical reaction and producing ATP. We need a lot of this. An active person can use 60 kilograms of ATP, can have a turnover of 60 kilograms a day of ATP molecules using ATP synthases. And that tells you a little bit how biology works. It's not just chemistry, it's chemistry coupled with electricity, with mechanics, and with everything else. So this thing is grabbing the ATP molecule, is twisting it, and by twisting it, it's actually making the electrons jump. So that's the key of the nanoscale. You can use mechanics to catalyze chemical reactions in like a machine. And this has the power not only to revolutionize the way we think ourselves, but also the way we create chemistry or the machines of the future. So I was part of the people that use the microscopes to start looking at these molecules for the first time. So this is, for example, DNA. DNA is not just a nice, pretty cartoon. It's actually a twisted molecule that moves, that feels the environment. That's why DNA created life on Earth, because it's able to entangle with the environment mechanically, electrically, and chemically to produce all the molecules. And you can see here the double helix that somehow is distracted by mechanics. But the most interesting thing of our microscopes is that allow us to see molecules moving in real time. So what I'm doing here is the highest speed atomic force microscopy image of the movements of a protein that is similar to the ones we have in our eyes, but also work in bacteria. So using these microscopes, the pioneering work of Noriyuki Kodera in Ando lab, working with Takayuki Uchihashi in the top with Hiroyuki Noji, we could see for the first time, for example, the movement of the top. Here's the ATP synthase looking from the top of what I was telling you before. Molecules grab other molecules, twist them, arrange, couple mechanics, chemistry, electricity. And perhaps the most beautiful thing is this here, made by Kodera-san, is a myosin molecule working on it. Sorry, yeah. So this is the an actin molecule a myosin molecule walking on an actin track. And this tells you a little bit how, again, why biology happens at the nanometer scale. So these molecules, you have them in your brain, you have them in your ears. Myosins are everywhere where you need some kind of contraction. And what life does is to create a little device like that. that is able to use the movement of the water molecules around them by a mechanical design to produce walking over the track. It also uses ATP, so it uses a combination of chemistry. Someone is talking in the background. I think someone should shut their computer. They're speaking in Russian and saying they don't understand. Okay, so yes, this is the link for me of physics, nanotech, and biology, at least at the beginning of my career. Let's move on. When we started to move to this area, the dominating idea about molecules, and still is now finally moving forward, was X-ray diffraction. In X-ray diffraction, molecules are put in a crystal, they're dead, they're outside their medium, and then you get the structures out of them. And it was, as you know, one of the big applications of physics to biology in the 1950s. I have put here Rosalind Franklin, who got the first experiments that led us to the destruction of DNA. And indeed, the hemoglobin and all these proteins that were. So with nanotech, we're bringing movement, we bring in the physics to all this story, but we also bring in an intellectual shift. So from the 1950s, most people were looking at biology as the building blocks. Remember the picture of the atoms? So everybody was looking at biology in the same way. We had X-ray diffraction. We were trying to identify all the proteins. There were dead proteins in pictures and cartoons. And we were trying to produce a picture of biology of an unimaginable complexity, where we were just having like a linear relationship between DNA and the proteins. And we were trying to simplify the complexity of biology by saying that life was a kind of algorithm written in genes. If we knew everything about the genes, we will know everything about biology. This idea of biology obviously collapsed with the Human Genome Project. We know that we have our DNA is much more than genes. We just have 5% of genes in our DNA, maybe even less. And now another thing we're learning is that genes collaborate with each other and that diseases are not depending on a single gene. But at the beginning of nanotech, we didn't know that. So we jumped into a new field, getting all these paradigms that biology, the molecular biology had for most of the 20th century. So we wanted to use nanotech in the same way pharmacology was using medicines. We were targeting individual molecules, we were targeting individual genes. And soon, like everybody else, we started to question this idea. And indeed, the first nanorobots that people were trying to imagine would be doing that, modify our DNA. So apart from physicists trying to understand molecules, what happened is that when we started to be able to produce nanoparticles, to monitor nanoparticles, to image nanoparticles, many people just trying to join forces with pharmacology to create better cancer treatments. I don't know if you know, but there are very few new drugs for treating solid tumors, especially in the last 10, 15 years. I will talk a lot about it. There's only been about 20, 40 new drugs. I have the numbers in a different slide, but they haven't been very effective. We still don't know whether they're very effective, is work in progress, but people thought that one of the reasons was that the drugs were not reaching, which is true, the tumors. That's why chemotherapy has to give you so much drugs. You need to concentrate it at the surface of the tumors. And Maeda, also in the 1970s, 80s, also found that nanoparticles can get through the vascular tissues, so to the vascularity that is around tumors. And that in that way, nanoparticles accumulate at tumors. And this was used to create a lot of drugs loaded onto nanoparticles to reach tumors. Unfortunately, there's only fewer than 20 nanomedicines that have been approved for use in cancer treatment, and they don't improve very much most of the outcomes of cancer treatment. Why? Because basically they were following the same mindset, the same intellectual framework of pharmacology. We just have proteins and we have individual proteins and we just target them with drugs. And what we know is that drugs and tumors become resistant. They're much more complex than our silly atoms or our silly reductions approaches. A cell can feel the environment, and the cell next to it understands it. They communicate with each other. They evolve resistant to your treatments. I will tell later how I think nanotechnology can help to improve that. One of the problems with nanotechnology as well is that the particles are cleared by our body. So when you put nanoparticles in our body, the kidney and the liver are able to get rid of them. So this is another of the obstacles we must reconsider when we use nanoparticles in medicine, which is still are not delivering the promise that we had. And I think the problem is that we didn't take into account the complexity of biology, and that looking for shortcuts is not fruitful. We need to move forward, be more multidisciplinary, and think deeper in our problems. Another area that nanotechnology has always been important, I'm going to check the time, okay, is been in biosensing. Again, initially, we were very naive. I remember when I was doing my PhD in Japan, people already had these pictures. Because molecules are at the nanometer scale, we thought, okay, now we can make, for example, here, transistors with carbon nanotubes, and then we can get proteins to stick to them with some chemistry, and then we will know what we have in our drop of blood, right? So the idea of nanotech has always been to produce what is now called point-of-care biosensing, in which we have a little device that is able to bind a specific protein in your body and tells you have the disease or not. And we are experiencing right now in the coronavirus crisis is that we are not there yet. We still rely in very, if you want, primitive ways of detecting the virus and detecting the antibodies. They work, but we could be doing much better. One of the reasons what nanotech has not delivered is, again, because it's a very multidisciplinary area. So engineers move in, talk to biologists, they didn't understand each other, and they found a common ground just looking at molecules, very reductionist. They forgot the physics of the whole thing. They forgot that we have very noisy systems. They forgot to talk to other people, maybe with people that understand more about complex signal processing. We're getting there. Some, for example, of the advances of sensing DNA with nanopores have come from machine learning, from interacting with areas of computing science which are focused on understanding patterns in complex systems. So what we are learning is, like, if we want to move forward, it's not going to be easy because the nanoscale is complicated, but we can make breakthroughs. And I think we are now in the point that if we form the right collaborations, we can make breakthroughs. Again, people are working a lot in that. I put an example here for one of the gurus of our field, Robert Langer, creating patches that we can stick to our skin and, for example, can measure glucose, and they can release insulin when you need it, not just by injection, which creates a lot of side effects. So all this is work in progress. It's been work in progress since the 1990s. And I think only because of the convergence of sciences, the breaking boundaries, being humble, and understanding complexity, that these are not simple systems, that we need to think very deeply, that mathematics and physics and computer science are needed to crack complicated programs, we can move forward. Now, let me go a little bit through the coronavirus. So, as I said, in our, like when you go to a conference in biosensing, you have things like this. Oh, we can create these amazing biosensors that they just communicate to our mobile phones, and we know everything about it. We know now that this is not true. What we are doing with the coronavirus, if you're lucky, is to get a normal PCR test. So basically, you get a swab, you get a virus, you need to amplify. This is a test that takes a lot of time, a lot of click of the pipette. It's very labor intensive. We don't even have many robots to do this. So there's a great need to create better testing. Again, ELISAs, we're getting better. We have a combination of, nanotech, very low tech, using nanoparticles, and trying to identify, amplify the signals, trying to detect the antibodies in the body. But one of the main problems we're having in the UK, and I think we're having all over the world, is that, for example, many people that has had COVID, this test don't detect the antibodies. We don't understand the biology of the antibodies. I think one great thing, which is anything good about this pandemic, is that it's forcing us to, as scientists and societies, to focus on reality. Our papers, our research is not just for papers and impact factor. We need to focus our collective scientific minds into solving problems to allow the survival of our societies. We're facing a lot of challenges in the 21st century. We will be having challenges with more pandemics and global warming and everything. We need to create more collaborative efforts to tackle the problems. And nanotech is at the heart because we need to detect molecules. We need to act on molecules. And my message again is just doing that is not enough. We need to understand the complex interactions between everything to be able to be effective. This is an area I've been working a little bit. Japan has invested in science of sensors for a long time, and I was lucky to be part of this collaboration with my old university, with Osaka University, and we just published a paper where we can see that field effect transistors made with graphene, when you are able to coat them with the right molecules, are very good for detecting. In our case, that was before the coronavirus, we were working with influenza virus, which is still a big problem, are able to detect viruses, are also able to detect the interactions of drugs with viruses, and people with the interaction of other problems to develop new drugs, to understand the kinetics, and they do it in real time. And the difference between these and PCR is that you just need a single drop on your device. You don't need many steps, because graphene's sheet is able to amplify the signal in a much more efficient way than the methods we're using right now to detect the viruses. Very soon after the onset of the pandemic, a group in Japan, I think no, this is in Korea, use a field effect transistor with graphene to detect coronavirus very efficiently. This is easy to fabricate. The technology is available. We need more work to get these devices for the general public and for the general use. But certainly there's a lot of things out there that can help to detect molecules in a better way. Let me talk about something else. So one of the aims of nanotechnology is to fabricate matter at the nanometer scale for creating drugs, for creating antibiotics, or for creating the materials of the future. I want to bring the attention to the work of the University of Washington that is led by David Baker. So the proteins are the building blocks of life. I just showed you a brief example about ATP synthase and the actins and how wonderful they are. Our collagen gives us our shape. Proteins are the most amazing materials that are on Earth, and we can assemble them into plants, into anything you can think of. So of course there's been a great interest in understanding how we predict the shape of proteins. For example, if we just know the DNA sequence, because it's so difficult to get crystal structures these days, it's a little bit easier with cryo-EM. So this was a parallel research that was a little bit far away from the things I was telling you before, but is now converging with us, which has been the field of protein folding. So people with computers for a very long time have been trying to predict how a protein, which is just a string, a nanostring, a polymer, if you put it in water, will have the shape it has. So this problem was intractable. There's so many ways in which a protein can fold that the computers didn't have enough computer powers to solve it. Yet people have been working for a really long time trying to solve this. So the team of David Baker and others have cracked the problem. Everybody thought it would be impossible to predict the size of a protein. How do they do it? Well, they did a combination of citizen scientists. They got a lot of people all over the world with computers helping them to do it, so they can share the computing power. But also they did a trick, which they used a finding from someone else, which was to include in the simulations to understand the protein structure, the history of evolution of that protein on Earth. So evolution, the constraints that the physics of Earth and the environment puts on us to live, creates the shapes that are good for survival. We are complex computers that allow us surviving. So he included this history, this evolution in the program. And by doing that, he was able to constrain the computation and get the right structures. One interesting thing in the context of the things I'm doing is, as soon as he managed to predict the protein structures using computer programs, he didn't stop there. What he wanted to do was to create proteins that didn't exist in nature. So he went to the computer, he designed the proteins, and then designed the DNA that would encode for these proteins, and went to cells and got cells such as yeast or bacteria putting the DNA inside to create these proteins. So created what is called post-evolutionary proteins, proteins that are built for their robustness. And he built them so they can fit like building blocks in bigger structures. This is a very radically new way of doing nanotechnology. This is not something you design with atoms in your head and then you just go in a reductionist way. Remember my picture of the atoms in the circle, and you build it. You take into account life, the complexity of life. You understand the evolution of life on Earth, and then you understand the kind of things you can produce using the power of biology to create. And then you go into the computer and you get life itself to create the nanomaterials of the future. This is very different from the idea that Eric Dressler had in the 1980s. And what we are learning is that if we want to create atomically precise nanotechnology, the best way is to use biology. So biology, nanotech help us to understand biology. And as soon as we do that, we get inspiration, like we always do as humans when we learn something about nature, to create something new, to create new materials. So David Baker is using these proteins and this ability to create, for example, virus-like particles that you can use as vaccines, new drugs, new materials. Indeed, there's a whole field of engineering biology. There is a sprouting, and it's a company called Cybergen in the US that is also in Seattle. I think they're in California and Seattle, actually. That is starting to use the power of biology to create the materials of the future. So I think the hinge of the phones that you have now, mobile phones that can hinge, are made of polymer materials that cannot be used by normal synthetic chemistry, but these days they can be produced by bacteria or by yeast. So we are moving to a moment in history. We're not building in a reductionist way anymore. We have understood that some things, and that if we move forward in our technologies, we have to forget that and we have to embrace biological complexity. Another case, for example, creating nanoparticle that kills bacteria. You know, bacteria have these problems that they evolve resistance to life. And I'm going to give you here a beautiful example. This is from the lab of Maxim Ryadnov in London, in the National Physical Laboratory, that he created a collaboration between computer scientists, physicists, mathematicians, biologists, and he created a little particle that is made of peptides. These peptides are chunks of protein that our own bodies produce to kill bacteria. So again, he's harnessing the history of our immune body in a nanoparticle. The nanoparticle is assembled from knowledge of how proteins interact with each other from physics, and is inspired by the shape of a virus. And as you can see here, doing again combination of mathematics and physics, very broad view, he has created, and his team, a little nanoparticle with ideas from biology to kill bacteria in a way they cannot evolve resistant to it. Similarly, Johanna Mella in Cambridge has created, with DNA origami, remember DNA nanotechnology at the beginning, little sheets with aptamers, again, pieces of protein that can sit on bacteria and kill them. One thing we are learning is that drugs or anything we use are much more effective killing bacteria when they are patterned in a similar way, like creating shapes, 3D shapes, that when they are by themselves, because they can harness physics as well as chemistry when they interact with the bacteria. Again, I'm going to talk a little bit about immunotherapies. If pharmacology has been really failing in treating solid tumors, not liquid tumors has been much more successful. Immunotherapy is the hope that we have to get rid of difficult tumors. Is this why? Again, we are working in the same way that we are working in making the new materials. Immunotherapy implies a very different approach to medicine. We are not just looking for single molecules, single genes. We use the whole body power. Our immune cell systems are complex, and they have evolved over millions of years to recognize cancer cells or pathogens. What we're learning is how to improve the immune response and how to make it stronger and control it. In my book, I predict that perhaps medicine will evolve a lot in the lines, at least a part of medicine, of creating a super enhanced immune system. So people are using, for example, nanoparticles to improve the treatment, the effectiveness of immunotherapy. I like this example very much. It's from many people are working on this. So they get a material, a porous material. We have a huge field, which is hydrogels, porous materials for drug delivery, who's been developing at the interface of material science, nanotech, and medicine for a long time, and so started to bring fruition. So for example, the group of these guys in Harvard, David Mooney, what they're doing is to use a material, and inside the material they put molecules that will recruit immune cells, and it will train the immune cells a little bit like the immune system to recognize cancer cells. I think we will have, in this case, for example, we will have vaccines that can be delivered for a very long time. One of the reasons we are worried now about, even if we have a vaccine for the coronavirus, which we don't know, it might just be effective for a little while because the antibodies will run out. So when we the problem is that we don't know enough about the immune system, but it is the possibility right now to create implantable vaccines that deliver these signals over a longer period. Some people are also working on injectable materials that we can put on under our skin and can help for cancer vaccine or whatever other vaccine. I'm going to finish a little bit more philosophically and reflecting a little bit more about the future of biology and where are we moving from now. So what I told you at the beginning is that physicists like me got interested in biology, trying to understand how molecules extract temperature, energy, chemistry from the environment to produce function. But also physicists like me are now moving on to something different, which is why did life emerge on Earth, and what are the at the nanoscale, which is something I told in the question size, and what interfaces are there with other things we're learning about nature, such as information processing, why biological matter predicts the future. I don't know, for example, plants are able to predict the weather. In Japan, cherry trees are able to change the color before the cherry blossom happens. We have adaptive behavior. We understand our environment and we process information, and then we get that information, for example, in the case of DNA, to produce proteins and adapt to our environment. How do we do that? And what does that tell us about, intelligent behavior? This is the new interface that is emerging in front of us. And I think, again, it will revolutionize the way we think about ourselves, material science and everything else. So I just put you here a picture of DNA vibrating. So how do I think about the origin of life, right? So as I think it now, and many of us think about, life on Earth emerged, or whatever it emerged, maybe came from somewhere else. When polymers, such as DNA, started to wriggle at the nanoscale, feeling the environment around them. So when the environment hits a polymer, like DNA, like RNA, or like any other polymer that is wriggling in water, it folds it. It changes its shape because the environment changes the way you are, the temperature, the water. And over time, that folding starts to, for example, be able to couple electricity or mechanics into the complex structure. And then something kicks in, which is Nonequilibrium Thermodynamics. If you start dissipating energy into the environment, you're able to become more complex, more order, to create higher order structures. You're going against this principle in nature that is entropy grows. Entropy grows unless you're hitting the environment, unless you're dissipating energy. And if to keep going, you have to keep dissipating energy. So if you keep going, you start to be able to put together chemistry, thermodynamics, and create more and more complex structures. These structures are in themselves information. They contain the information of the environment, the kind of information that lets us persist over time. Of course, we don't know how you go from the level of a molecule to the whole cell. This is one of the main problems of understanding the origin of life. But combinations of physics, non-equilibrium thermodynamics are leading us to understand better the coupling between information, materials, biology, and life. And I think that this interface is where, okay, I put this here, what I said at the beginning, it's going to bring us to a new revolution in what we understand. We will revolutionize computing, materials and everything else. We're just scratching the surface with machine learning. I don't know if you know, but machine learning, people try to translate. I used to be very interested as a student in translation. Our algorithms, when they were reductionist, when they were just top down, they didn't manage to translate languages or to recognize patterns in pictures. Machine learning became success when it implemented something we call artificial neural networks. That means the algorithm somehow mimics the structure, the layer structure, the nested hierarchical structure of the neurons in our brain. What we have learned from machine learning is that when you have structures that are built hierarchically in complex structures that resemble biological structures, we suddenly start to understand the world around you, or at least recognize it. That's what machine learning does. In a way, I was telling you before, the biological molecules are storing information in themselves. So the next step will be to see how biology achieves this, which is what I was trying to do. I'm not sure this is going to play. Let me show you this video. So another phase, another area that is starting to interface with nanotechnology, material science, medicine is the phase of natural intelligence in simple systems. For example, what you see here is a slime mold. It's a little mold that grows on wood. And what people found out that just because of the constraint, the way the mold grows on itself, it can't compute. In this case, I don't know if my YouTube is going to grow. What you see is that the mold is growing, and they have some little hubs of food in the areas where in the map of Tokyo, the main stations of Tokyo area. If you let the mold grow, just because of the constraints of the biology that makes the mold grow, it will compute accurately the network of the underground in Tokyo. This is again the interface between computer and materials, non-equilibrium thermodynamics, and a structure that will shape our future. Oh, something happened here. Okay, and another example, just to finish. This is an experiment from Jim Jimsevsky in UCLA. He used to be an atomic force microscopist, can entire microscopist. What you see here is a network of nanowires of silver. And what he has put is a lot of interconnections between this network of nanowires. So basically, all he has produced is a highly interconnected nanowire network, a little bit like machine learning. You just try, but in the physical world. The question is, can highly interconnected matter actually learn about the environment? So what he did is to put this on a bed of electrodes, and he started feeding electrical signals on one side that were telling this, that the signals were like information about the traffic in Los Angeles. He lives in Los Angeles. And he let the system go. Because you're so small at the nanoscale, the electrical signals are changing you over time. It's changing you over time. So you let it go. And after some time, it just came back, and the system started to measure the signals in the other side of the pad of the electrode. And funnily enough, the system was starting to predict the patterns of the traffic in Los Angeles, because complexly interconnected matter has the capacity to understand the environment. There's nothing, what we have done with computing is to separate the physics, the physicality of learning from the physical world. But actually what we are reconnecting now with computing and understanding how matter at the nanoscale, when it's highly interconnected, learning from biology can learn. This can learn, and who knows what we can do with it. So, and I just finish with a little bit more. Harnessing this capacity of biological systems to understand the environment is also poised to revolutionize tissue engineering. So I don't know if you know, but if you put a cell on the surface, depending on the surface it will stick, it will pull from it, it will understand its mechanics. In the case of a stem cell, it can change. If you put a stem cell on something as soft as brain, develops over time into something similar to a neuron. And if you put that stem cell into something hard as bone, it develops into something like bone. So using this capacity of cells for understanding information, processing information, and changing and responding to it, people are starting to create materials that can help to regenerate our body. Of course, the most simple way to do this is to transplant in some material from someone that is dead. So for example, you can, if someone loses a trachea, you can get a piece of trachea to someone that is dead, remove the cells, get the collagen, seed it, and then you get something to grow. But of course, the most interesting part is if we can do this with artificial materials. So people are starting to make materials that help to reconnect neurons after spinal cord injury. I don't have time for this. One of the most interesting developments is to create little organs that can go in chips. Of course, they're very tiny because it's very difficult for us to produce in tissue engineering a big organ. Is something I discuss at length in my book. But what people are starting to do is to use these little organs to connect them with each other and to test drugs. So I think the first, instead of using animals. So this is a bit, I'm going to finish here a little bit. So I think the future of medicine will be a combination of very good sensing, collecting real-time measurements, probably of the nanometer scale, proteins, probably cells, combining both reductionist approaches with emergent approaches, approaches that are simple, looking for individual molecules, but also approaches that are using the complexity of biological system, using cells, using complexity to understand the body. We will have increasingly smart mathematical model that combines everything, that combines our knowledge of physics, of emergence, of complexity, and machine learning, everything in increasingly sophisticated computer programs that are able to read big data, but not just big data with machine learning. That will not tell us what is going on. We need big data with machine learning and proper understanding of what is going on. That's going to come from physics, from profound philosophical and hard thinking in mathematics and physics terms about what is the meaning of the information we are getting. Machine learning alone will not tell us what is going on in our body. Machine learning is a tool, and we need to harness it with the rest of our mathematical and intellectual tools we need to understand the body. So somehow with all this, we're getting closer to the dream of traditional medicines, which is something we always tried as humans. So, you know, in Chinese traditional medicine, I'm sure you have the equivalent in Turkey. We had these holistic approaches to disease, in which you get the pulse, for example, and you're trying to understand what is going on in the whole body, and you have a human that has been trained over with the knowledge of many generations to understand disease. This is very similar, but we are now trying to use modern science to try to reach the dreams of traditional medicine. And of course, I finish here, and this I think will be the last slide. With one of the predictions I do in my book. So we are moving to a future where we start to see less boundaries between biological and physical sciences. And this is a beautiful example. So this is done by Kit Parker at the Harvard Wyss Institute, where he created a toy. It's a toy made of plastic, of polymer, and sitting on it, there are heart cells from mice and electrical signals. And the toy is able to swim a little bit like a stingray, and it's able to follow signals. These toys, we will learn how toys learn, how our brain learns with these toys, how the materials of the future, a new world of applications will come out of playing at the interface of all these areas. I think, I don't know, this is finished. I work, just one slide to think the sort of things I work with. I'm interested in plants, the intelligence of plants, how plants evolve, grow, and create their shape in response to the environment. I use a combination of experiment with atomic force microscopy, polymer physics, biology to understand how plants grow and react to the environment. I also work in mechano-electrical coupling in neurons in the context of ultrasound neuromodulation. We know that ultrasound can change and can help conditions such as epilepsy, and we're trying to understand how we can use ultrasound to change the electrical signals in the body. I also create biomimetic materials for tissue engineering, drug delivery. I'm also working on the physics of pancreatic tumors, trying to understand how can we enhance the delivery of drugs and the behavior of pancreatic tumors. And I also work a lot in cryopreservation. I want to understand the physics of freezing. And with this, I think I finish here my presentation, and I will take questions from.
Yeah, okay. Well, thank you so much, Professor Contera. You know, Professor Contera, you can hear me well? No?
I can hear you.
Yeah, okay. Well, Professor Contera described exciting ways of nanotechnology. The future will bring synergy of nanotechnology with biology, medicine, and cutting-edge fields like robotics and artificial intelligence. At this point, I would like to mention about the project that we are running in Sabanc University Nanotechnology Research and Application Center, and I would like to give floor to the Fazilet Professor Vardar. Maybe she can explain a little bit about our projects.
Very shortly, actually, it's sort of peripherally related to what you're doing, but it's a virtual cluster of sensors, biosensors. And we've got 12 academic institutions and, 8 academic institutions and 16 industrial companies in this virtual cluster. It's a platform, and we will be conducting 17 different projects in biosensing of agents that cause infections or diseases. So it is the monitoring and of health parameters, wearables and biosensors and biochips. The basic point that we may be searching for partners would be this complex signal sensing that you are, because we are very good in creating
engineering, right? Maybe.
Yeah, it is we're good at creating the functional surfaces and getting a recognition of the agent that we are trying to sense. But how can I say, interpreting or translating it to a signal that is free of noise.
This is the biggest channel. Biological systems are very noisy. We have a lot of information. And what biology does is to reduce the noise by creating a structure. So the way immune cells, I always think that it's very useful to understand immune cells, or trying to think how any biological system thinks. So immune cells, when they find a pathogen, they grab it, they taste it chemically, they have a lot of receptors, they squeeze it, they feel their mechanics, so they do taste and texture. We are very far from that, because the thing is, the immune cell is a complex computer in itself. So it gets all the signals, and through evolution, it's creating all these roads that links to DNA, and the cell computes and say, boom, pathogen out. Or the problem is when it doesn't detect it, because viruses also evolved to remove that. So I think this is a general problem with biosensing. We don't crack it. We are far from cracking it, because I think we don't have the right collaborations. And I think maybe in Turkey, you have a good opportunity. You're very good at engineering. You're very good at computing. I think we need as a community to collect more people from computing of complicated signal processing, trying to understand what our problem is. And this communication is very complex and takes a long time. But I mean, this is the main problem we have in our field. I mean, how do we sense complex signals in high noise? But again, computing is learning a lot from this. The development of machine learning, artificial intelligence, they're trying to do the same with all the type of problems, usually to make money in some markets or something. So maybe we can use this way they're doing to improve our medical devices. I think we need a collective effort for this. We need to convince the right people and have vision about this. I think the pandemic gave us an opportunity to think more. I think we need to think more. We need to be more honest, and we need to find our weak points, and then and try to move forward. We're all doing the same. We all build our devices. And now we are good at detecting single molecules. Before we were not. Now we are. But now we need to create the devices that make a difference. Who knows? Maybe even many different approaches. But we're all working on it. I think probably in Turkey you are in a good position to move some ideas. So you need to be strategic maybe and see, focus what are your strong points? I think you have a lot of strong points. Maybe you're better situated in many ways than many European countries. So yeah, I very much encourage you to speak to people of computing and signal processing, and to think carefully. I think we need to be critical in the way we do science, because we progress very slowly, especially multidisciplinary science. We've been doing multidisciplinary science already from the 1990s. So I think we need to move to the next step. We need to be more critical with ourselves. Where are our weaknesses? It's great in Turkey, you have a lot of women in science, and I think that helps to multidisciplinary research. I think we're more used to tackle complex problems. Again, having diverse teams helps. So what I see in your audience is also very diverse. So actually, you are very well situated.
So we would really appreciate if you could connect us to.
I will. I am going to.
Who might be interested.
I want to write. I'm writing right now, or thinking of writing right now, a proposal, or trying to think carefully about what is the best way to create sensors based on field effect transistor, for example, and that really make a difference. And I am talking myself to engineers that are interested in signal processing in noisy environments. So I can contact you and we can join forces. I will do it offline. I will do it offline, and I will be very much happy to share our information and see if we can, maybe we can go for broader European networks or whatever we can do.
Yeah, we would really like that.
Actually, yes. I'm talking in the European Union next week about these things. They invited me to give a seminar about nanotech in medicine, so they are interested. So let's try to push this and see if we can bring better sensors, because sensors is needed for understanding everything around us. We need better data.
And Turkey as a partner will contribute to the diversity.
I'm sure you will. Well, I also contribute. Turkey is great for women in science. It's one of the most wonderful things.
Thank you.
Well, here is one question from the audience, if it is possible, okay for you, Professor Contera.
Of course, I love questions.
Yeah. How well do we understand the role of water in biological complexity? Is a biologically active environment in the absence of water molecules possible?
No. No, because it can be something that is no water. We don't know that. I mean, one of the things you learn when you do nanotech, remember our pictures of the atoms and the surfaces. So is that when you're so small, you feel everything around you. The environment totally changes who you are. So one of the reasons why life is possible is because the cell creates a clean environment for the molecules to work. The cell totally controls what is inside. It's clean water with salt in the right concentrations. Evolution has made sure that you have the right concentration, and the cell membrane is full of sensors much better than ours, to understand the right concentration of potassium, the right concentration of sodium, because if it just goes a little bit off, it doesn't work. Many diseases come when you have potassium channels malfunctioning. So so if you want to have nanomachines working, you need to control the environment totally. You can do it two ways. You can do an ultra high vacuum, so I guess in space, or you can be in a liquid that is controlled. So life on Earth emerged on water. The reason is because there was water, but because water allows many things. We still don't understand the physics of water, the structure of the interface. So a protein or DNA is itself, but only because of the salt and the water around it. Actually, there's only 3 molecules of free water between 2 molecules in the cell. All the water is bound. It's part of the structure. It's part of the way everything works. Everything is interconnected. Biology emerges from interconnection, not for individual work or individual blocks. And water is fundamental in the whole story. And we don't know enough because it's very small. It's difficult to model. And it is, of course, work in progress, how we integrate everything with a picture of water.
Well, thank you. Well, here is the last question for you, Professor Contera, from the audience. Is it essential for a particle to have a dimension less than 100 nanometer that we mention it as a nanoparticle? In some techniques, there is difficult to reduce the dimensions less than 100 nanometer as in electrospinning.
No, depending what you want to do. I think the 100 nanometer is a definition that was actually made by the European Union. At some point, people needed to legislate on the safety of particles. We need to make laws, and we need to define what is a nanoparticle. There was a big commission, and everybody did, and the agreement was that we would consider a nanoparticle something that in one of these dimensions is smaller than 100 nanometers, I think. But for practical applications, you don't care how you call things. If it works, if it's 150, you make it 150. Look at viruses. Please get inspiration from viruses. Viruses come in the nanometer scale from 100 to 200 nanometers, 1 micron, and they just adapt their shape. So if in electrospinning you do bigger particles, it's fine. We'll find something to do with them.
Well, Professor Contera, we are running out of time. Would you like to add anything before we finish the webinar?
No, it was lovely to meet you all. I would like to hear more from your activities. I think I'm going to get in touch with the people that kindly invited me. Yes, and I'm open to questions if you want to do. I'm on Twitter, and I'm happy to reply to questions, and I'll be in touch regarding biosensors. I am very happy to hear your interest in biosensors because I think it's a key area where we all need to work much more. It can help us in medicine, but also can help us even intellectually to understand biology. So I'll be in touch.
Well, thank you. Thank you, everybody, for joining us today. Take care, keep safe, and stay healthy.
Lovely to meet you.
Yeah, likewise.